All right, we are going to be covering cell transport in a series of 2 videos. So please make sure that you're watching carefully, and this should help you with your notes on page 29, 30, and 31. Before we talk about the process of cell transport, we need to talk about the structure of the cell membrane. This here is an animated picture of the cell membrane. I did ask you to draw one in your notebook, and this might help you understand what those parts are. Number 1 here refers to the cell membrane and everything else that's inside of it. Number 2 refers to proteins. We'll talk about the function of these proteins in just a little bit. Number 3 refers to the lipid bilayer. Bi means 2, so bilayer means that there are 2 layers of lipids that compose the cell membrane. Number 4 refers to these things that are sticking out of the cell membrane, and these are carbohydrates. Number 5 refers to a protein that has a passage channel in it, and this is called a transport protein. And then number 6 refers to a phospholipid, one single phospholipid. If you multiply these over and over and over again, you will get the entire composed cell membrane. The function of a cell membrane is to regulate what enters and leaves the cells. It lets good things in, gets rid of waste. It also provides protection and support. It allows things to stay inside of the membrane that need to stay in, and allows waste to leave. Again, it is made up of a lipid bilayer, 2 layers of lipids that contains proteins, and it's also what we call a flexible structure, and it's a barrier between the inside and the outside of the cell. You can see here that my phospholipids here are moving. This is going to be what the entire cell membrane does at all times. It will constantly move back and forth like this. You might remember this from our bubble example when we set a rubber band on top of a bubble and then you saw it move. The cell membrane has proteins that are here in pink that will form channels and pumps to help move things across the cell membrane. You also have these blue things here that are called carbohydrates, and all these serve to do is to identify different cells. They basically are a communication mechanism to allow cells to talk to each other. Cells are maintaining homeostasis at all times. The definition of homeostasis is a balance that organisms maintain through self-regulating adjustments. This is a good definition to have somewhere in your notebook, probably the bottom of page 29 if you have room. A cell is an open system. This means that things will go into the cell, such as matter and energy, and things like waste will leave the cell. It's kind of like a little living organism. The cell is what we call selectively permeable or semi-permeable. These phrases are used interchangeably. They mean the same thing. It basically says that it will let some substances pass through more readily than others. Okay, it's kind of like a bouncer. It's going to let things that should come in and keep other things out. One of the most important things that you need to understand when it comes to cell transport is going to be the concentration gradient. A concentration gradient means that in the absence of other forces, materials will always move from an area of high concentration to an area of low concentration. I want you to think about perfume and cologne for this example. When you spray perfume, right next to you, there's a high concentration of those perfume molecules right next to you. Eventually, those perfume molecules will spread out, and maybe somebody on the other side of the room will be able to smell those molecules. This is taking those perfume molecules from a high concentration to an area of low concentration on the other side of the room where there were no molecules. We're going to look at a diagram that will help you understand this. So in this diagram, we're going to be labeling a couple of things. The first thing we need to label is the cell membrane. This is going to be the barrier. Here you can see that there is an area of high concentration. Okay, there's a large percentage of these purple molecules. And here there is a low percentage of these purple molecules. Because of our concentration gradient rules, things are going to move from an area of high concentration to an area of low concentration. So these molecules from the left would simply move over to the right. We're going to look at another diagram that's very helpful for this. In A, you see a high concentration of some solute molecules on one side of the cell membrane. Next, in B, you see that these solute molecules are naturally moving from the area of high concentration to an area of low concentration. This movement is going to continue until the concentration is equal on both sides of the membrane. Finally, in C, you can see that what we have reached is called equilibrium. This means that the concentration on both sides of the cell membrane is equal, even though the concentrations are finally equal by that movement from high to low. What will happen is those molecules will still consistently move in both directions, but the concentrations will still remain equal. An important takeaway from this is even in equilibrium, we still have movement of those molecules. We're going to talk about two types of transport, passive and active transport. The rest of this video will cover types of passive transport, and the next one will be about active transport. In passive transport, okay, this is again involving molecules going across the cell membrane. In passive transport, no energy is being used to move these molecules across the membrane. They are naturally going with the concentration gradient from a high to low concentration. One of the definitions that you need to have on page 29 is the definition of diffusion. Diffusion is the spreading out of molecules from an area of high concentration to an area of low concentration. The perfume example that I gave is a perfect example of this. We're going to look at some scenarios that are problems that you will need to be able to solve, and some examples are in this video. In this beaker, we have some water, and in the water we have a bag. The bag is going to be 20% salt solution, and then the water is a 5% salt solution. I'm wondering in what direction the salt molecules will move. If I know that in diffusion, molecules go from a high concentration to a low concentration, I'm going to look for my higher number, which is 20, and I know that because things go from a high to a low concentration, my salt will move out of the bag. So again, the salt will move from an area of high concentration, 20%, to an area of low concentration, which is my 5%. Another type of passive transport that you need to know and be very clear on the difference is osmosis. Osmosis has a very similar definition to diffusion, but osmosis is simply the movement of water across a membrane from an area of high to low concentration. You'll notice that the lab that we did with the potatoes was an osmosis lab. We saw the movement of water. Please be very clear that osmosis is only dealing with water and diffusion is any other molecule. We're going to look at the same situation. We have a bag that is 20% salt in a solution that is 5% salt. We're going to need to do a little bit of math. Since we know our salt solution percentages, we can figure out our water solution percentages. If we take 100 minus 20%, we get 80%. So we know that the bag is 80% water. If we take 100 minus 5%, we get 95%. So we know that the outside solution is 95% water. Again, things move from a high concentration to a low concentration. In what direction will the water move? I know that it will move into the bag because it's going from this high to this lower concentration. So water movement will go into the bag. There are 3 types of water solutions that we need to know. Whenever you see these words, you are still dealing with passive transport and the transport of water only. There are different types of solutions that cells might be in, and you need to know what happens to the cells in those types of solutions. In an isotonic solution, the amount of water is the same on the inside of the cell and on the outside of the cell. This is an ideal situation for cells. They want to be in isotonic solutions because they are at equilibrium. Water will flow in and out at the same rate in both directions. This is what we call dynamic equilibrium. In a hypertonic solution, there is going to be more water on the inside of the cell than on the outside of the cell. And since things will move from a high concentration on the inside to a low concentration on the outside, my water movement is actually going to be out of the cell. Okay, this is my animal cell here, and this is a plant cell here. Notice that a plant cell will kind of stay the same because of that cell wall. Only the cell membrane, which is here, will shrink. Okay. Sometimes, because too much water is moving out, we go through this thing called plasmolysis. It will mostly just refer to animal cells because they don't have that cell membrane. Too much water can move out and the cell can collapse. In a hypotonic solution, there is going to be more water on the outside of the cell than on the inside of the cell. So since we're moving from a high concentration to a low concentration, the net movement of water will be into the cell. Notice my animal cell looks different than my plant cell. Sometimes we can go through a process called cytolysis, which is when too much water will move in and the cell membrane will burst due to water pressure. This is called lysis. Okay, so lysis is when a cell bursts. So animal cells can burst. Plant cells don't often burst. Okay, they have a cell wall. Because of our cell wall and our central vacuole, what happens is the plant cell just becomes extremely full of water. If you think about a plant that hasn't been watered for a long time, it's wilting, right? It's kind of bent over, doesn't have any life to it. If you add water, okay, if you're adding water to a plant, what's going to happen is that large central vacuole is going to fill up, and what that's going to do is cause this thing called turgor pressure to increase. That just simply means that there's a lot of water in here, and that plant will start to perk up and stand up, and it will form these very tightly packed cells, and that plant will be kind of alive again. This will help give some structure and support to our plant cell. The last thing that I want to show you is going to be something that I'm going to want you to remember, and we're going to talk about it in the next video. This is Drake. Yep, that's Drake. And what he's moving are some molecules across a membrane. We're going to talk about what this means in our next video, but here is our cell membrane. This is the outside of the cell. This is the inside of the cell. Drake is moving some things from this way and back this way. Okay, we're going to talk about this in our next video. I do hope that this helped a little bit with some cell transport.